we are not so familiar with iron 6. Iron 6 is a high-valent iron oxo species. And the reason that is very interesting is that it's a very powerful oxidant, so it can be used to oxidize organic and inorganic contaminants in water. It's also a very good disinfectant. We can inactivate bacteria and viruses. And another feature of iron 6 is that after its application is reduced to iron 3, and iron 3 is a common coagulant in wastewater treatment. So basically we can have 3 actions with one chemical, and this is oxidation, disinfection, and coagulation. And the people that started investigating iron since the last 25 years in the water treatment area, investigating the oxidation of different chemical pollutants and the inactivation of biological contaminants in water and wastewater. So here is a table with the redox potential of the most common oxidants, disinfectants used in the wastewater treatment. And here is hypochlorite, ozone, hydrogen peroxide, and dissolved oxygen. This table has two main messages. The first message is that the redox potential of an oxidant is a function of the pH. So this is very important when we talk about oxidation processes. Each oxidant has a different redox potential in acidic and in basic conditions. And the second message is that iron 6 has the highest redox potential in acidic conditions among the most common oxidants in the wastewater treatment. So it really makes sense to investigate this oxidant and try to see if we can take advantage of this high redox potential for wastewater treatment applications. So here is a speciation of iron 6 as a function of the pH, and here we can see that at lower pH we have different iron 6 species, and those at lower pH, those species are more reactive than the species in the high pH, and this is how we explained the higher reactivity of ferrate 6 at different pH. And now, before going to the experimental details and the results, I would like to talk about some fundamentals about ferrate 6 that it will be very helpful to understand the findings of this experiment. So I'll try in 4 sentences to give you a nice overview of some very fundamental things about iron 6. So the first is that the oxidation power of iron 6 depends on the pH. This is what we saw. This is true not only for ferrate 6 but for any oxidant. But specifically for these findings, this is very important to remember. Another thing is that iron 6 reacts with water. So if we have iron 6 alone in water, it's going to decay to iron 3. The third one is that in acidic and neutral pH conditions, iron 6 has high reactivity. As we said, there is a higher redox potential in acidic conditions, but also the reaction with water is faster. So there is a competition of reactions. At the low pH, the redox potential is higher, but then the reactivity is higher not only with the pollutant, but also with the water. And when iron 6 is going to react with water, then there is not available to react with the pollutant. So there is a competition in the kinetics in this case. And then at the slightly basic pH conditions, around 9 to 10, where ferrate 6 is more stable, there is decreasing reactivity. The redox potential is lower. So having in mind these four sentences, there was a clear gap here, and ideally what one would do was to try to find a method to activate or catalyze iron 6 oxidation reactions at slightly basic pH conditions, where iron 6 is more stable but has lower redox potential. And obviously by doing this, we would like to have higher removal of the organic contaminant, and then at lower concentration of iron 6, and of course at lower contact time. That would be an ideal scenario for the iron 6 technology. So we at the initial part of the experiment, we selected few organic compounds as model compounds, and here I'm showing the structures. Is caffeine, which is a psychostimulant, is acesulfame, potassium is artificial sweetener, and Atenolol is a beta blocker. I'm sure most of you are familiar with these compounds. They are frequently detected in water streams, and people investigate their oxidation by different oxidation processes. Yeah, it was also important to choose some compounds that they don't react very fast with iron 6, because we would like to show the activation or the enhancement. So if the reaction was very fast, that would not be possible. And these three compounds, they did not react very fast with iron 6 without activation. So it was ideal model compounds for these studies. But that was something we realized after we performed the experiments, and you will understand what I'm talking about soon. So here is the experimental design. It's a very simple experimental setup. We produce iron 6 in the lab using a wet chemical oxidation method, which I'm not going to talk a lot about it, because there was nothing innovative here. It's simple oxidation of iron 3 to iron 6 using another oxidant. So this powder, the iron 6 powder, was added in milky water. Then we centrifuged to remove some iron 3 impurities. We could standardize, we could measure the concentration of iron 6 using a UV visible spectrophotometer. Iron 6 has a very characteristic purple color and absorbance of 510 nanometers. So it's easy to quantify the concentration. And we had a solution with the model compound. In this case, is example of caffeine. And we added iron 6, and then we could add the activator. In this case, I'm showing an example of the acid. And then at the end of the reaction, iron 6 is reduced to iron 3. We can filter and remove iron 3, and then we can measure the model compound using high performance liquid chromatography technique. So that was the experimental setup of most of these experiments that I'm gonna present today. And now I will go through some results and how we design experiment by experiment when we got some unexpected findings, and then this ended up to the discovery of activation of ferrate 6. So in the first set of the experiments, we had that experimental setup. We had iron 6 and caffeine in the solution, and then we've been adjusting the pH using hydrochloric acid during the experiment in order to maintain the desired pH. And here we can see a nice curve that shows that different pH did not affect the removal of caffeine in the solution. And we can see that the removal of caffeine increased with increasing molar ratio of iron 6 to caffeine, which was something that we expect. Here in the inset figure, we can see the decomposition of iron 6 in the presence and in the absence of caffeine. And here I'd like to say that the last 20-25 years, most of the kinetic studies on the iron 6 oxidation reaction was made by monitoring iron 6. As I said, iron 6 has a very characteristic purple color. Then when a researcher wanted to study the kinetics of the reaction of iron 6 with a model compound or a model pollutant, then we could monitor the iron 6 and using a spectrophotometer, and then we can study the kinetics of the reaction A plus B, where A could be the iron 6 and B could be the pollutant. And when we try to do this type of experiments, in this case of the reaction of caffeine, we use the initial rate method, which was one of the most commonly used for iron 6 reactions and many other reactions. And is still used a lot by reaction engineering researchers. And then initially, because we've been interested about the initial rate of the reaction, if you see on the left of this red arrow, there was no difference in the iron 6 decay with and without caffeine. So that was a surprise to us, because that shows that there was basically no reaction between iron 6 and caffeine. However, we had performed experiments before, and we saw that there is a caffeine removal when in the presence of iron 6. So these two results were contrary and really unexpected, and we decided to perform some more experiments, having in the solution only iron 6 and caffeine, without any acid, without adjusting any pH, to see if if caffeine is going to be removed, and these are the blue bars here. And we can see an increased removal of caffeine when we increase the molar ratio of iron 6 to caffeine. And this was very interesting to us because kinetic studies based on initial rate, we didn't have the picture on the right yet. So we had only the picture on the left side of the red arrow. The two figures were, the two studies were contrary to each other. And then when we look more carefully the results, here, for example, at molar ratio of 4, we can see 40% removal of caffeine. But if I go back to the first figure that I showed today, at the molar ratio of 4, we can see 60% removal of caffeine. So here we said, okay, something is interesting here. When we do the experiment and we adjust the pH with the acid at molar ratio of 4, we can see 60% removal. Then when we do that without the acid, we see 40% removal. And when we do kinetics based on the initial rate, we see no reaction. So said, okay, so we need to go deeper and investigate more. And then we designed some new experiments to see if the difference in the experimental design between these blue bars here and the experiments that I showed before, there is any enhancement because of the acid. Then when we increase the concentration of the acid, we should see some increment in the removal of caffeine. So in this case, we started measuring the amount of acid added in the solution. And we can see here in the figure A, at different molar ratios of iron 6 to caffeine, when we increase the concentration of acid, we see an increment in the removal of caffeine. That was really interesting at that time. And then to figure out if this increment is due to the H plus ions of the chloride ions, we use another acid, the HNO3, and we saw a similar result. So we've been confident by that time that increasing the concentration of acid, we can increase the removal of the contaminant. And then we try another organic acid, the acetic acid, and we saw something similar. And then that was very fascinating because we basically could activate or catalyze the reaction using simple acid, and we could get complete removal of the contaminant at a lower amount of iron 6. And here I'm going to give an example. This is the removal of caffeine after complete reaction at different iron 6 to caffeine molar ratio. And without activation of iron 6, we need a molar ratio of around 25. But by activating iron 6 with some micromolar of hydrochloric acid, this molar ratio is around 8. So we are talking about 3 times less amount of iron 6 needed for the same removal of a model compound. And that was really exciting. And but if we remember what the fundamentals of iron 6, I said that the oxidation part of iron 6 depends on the pH. And then the first thing that the first thought was that this is happening because we have a decrement in the pH. So at lower pH, we expect a higher removal of a compound. And this is what is going on here, and there is nothing new basically because that was known. But if we take a look in this table, I know there are lots of numbers here. But if we take a look in this experiment number 6, we see that from an initial pH of 9.5, the immediate pH after the addition of acid was 8.8, and then the pH went back to 9.1, and then the final pH after the reaction was 7.5. So basically, we didn't have any drastic decrement of the pH by the addition of acid. But we could get a very short contact time to achieve a complete removal of the compound. We are talking about seconds compared to hours without the addition of acid. So these pH measurements, they showed that this finding was not related to the pH change, but it was something new. But then we performed further experiments in these slight basic pH conditions from pH 8 to pH 9. And here we showed that these findings were not related to the pH change, but they were related to the activation of iron 6 using simple acids. And to make sure that there was no interaction between the acids and the compound or the iron 3 produced by the decay of iron 6 with the compound, we went through many control experiments. Basically, we try to do any possible experiment to rule out all the possibilities. We ran control experiments with the different acids alone and caffeine, and we saw no removal. Then we designed some new experiments with iron 3. What we did, we got iron 6, and we let iron 6 to reduce to iron 3 in the presence and in the absence of acid without any more compound. And then we use that iron 3 solution to oxidize caffeine, and we received a result with no removal of the organic compound. So this after doing all these control experiments, we knew that there is no interaction with caffeine and acid. We knew that the iron 3 form at the end of the reaction also had no role in enhancing the oxidation of caffeine. But still, we had the question why our kinetic studies, this is the instant figure that I showed at the beginning, why when we have, why the initial rate was different, then we repeat this experiment and we let the reaction for longer time, and then we saw what can be seen at the right-hand side of the red arrow, which is a differentiation between the decay of iron 6 in the absence and versus the presence of caffeine. So these then these experiments, they really close the loop of this work, and they show to us that there is a reaction between iron 6 and caffeine, and this could be seen from this kinetic study here, and the difference between these experiments and the first figure that I showed in this webinar was due to the addition of acid, and the control experiments and the effect of the pH experiments, they could prove that this was an activation of iron 6, or that was something new and not a simple effect of the pH that was known by that time. So it was really exciting to go through these experiments and design new experiments to investigate this system. And I'm sure that many of the PhD students here in this webinar today, they went through something similar with some unexpected results. And then when this is a time when research gets very, very exciting, and we really try to figure it out and find something new. So the next question we had to answer was, what is the reason of this finding? Why iron 6 is activated in the presence of acid, and what would be the species responsible for this observation? And to investigate this, we perform some experiments in the presence of nitrogen. So basically, when we do an experiment in the laboratory, we have approximately 9 milligrams per liter oxygen that is dissolved in the solution, and then what we did is that we purged nitrogen to remove this oxygen, and then we performed the experiment in the presence of nitrogen. The reason we did that is that we knew that there are two possibilities in this system. The one was that the enhancement is due to reactive oxygen species, and the second was that this enhancement was due to iron-based species. And by doing the experiment in the presence of nitrogen, we could somehow exclude the one part, which was the reactive oxygen species, and we could conclude that what we observed is due to the iron-based species. And here, these results, they show they showed almost no difference when we purge nitrogen, and this was an indication to us that the observed enhancement in the removal of a model compound by Ferrate 6 was due to iron-based species and not due to reactive oxygen species. And considering that we knew that there is no effect of the iron 3 in this system, and we also measured the effect of the iron 6 alone without any acid, the two species that are between the oxidation state of iron 3 and the oxidation state of iron 6 is iron 4 and iron 5. So we could conclude that considering that we could show that this enhancement was due to the iron-based species, then the oxidation state of this iron species would be iron 4 or iron 5. And how this would be produced in this system, we can see here, for example, one is 1 or 2 electron transfer from iron 6 to form iron 5 and iron 4, and then ultimately iron 3 and iron 2. So it seems that the acid is enhancing the formation of iron 4 or iron 5, or it makes iron 5 and iron 4 more stable to react with the model compound. And this is what is shown here in the second set of reactions, that iron 4 and iron 5 could react with the model compound rather than iron 6. Also, all the high valent oxidation states of iron could self-decompose. So maybe the acid retard the self-decomposition of iron 4 or iron 5 but enhance the decomposition of iron 6. So all this may happen at the same time in a reaction system like that, but all these are possibilities to occur. And finally, it can be reactions between the iron species between them to form iron 4 or iron 5, and also some interaction with the oxygen species, which is most likely this is not the case based on the experiments we did in the presence of nitrogen. So after having all this conclusion with the proving that we could activate iron 6 using simple acids to enhance the removal of caffeine in water, we expand the study to two other micropollutants, to Atenolol and Acesulfame potassium, to make sure that these observations and these findings are not related to a specific model compound, but can be expanded to other organic contaminants. And here we can see that in the case of ACE, we had very similar results with the case of caffeine. So by increasing the concentration of acid, the removal of ACE was enhanced. And in the case of Atenolol, iron 6 alone, without any activation, basically did not really react with Atenolol. But by activating iron 6 using some amount of acid, we could have some removal around 30%. And what was interesting is that the enhancement in ACE was around 30%, 35%, and then also for Atenolol, this enhancement was also around 30%. And what was important is that this was happening in seconds instead of hours. So basically, the contribution was that that was something new. That was something that was not known the last 20-25 years that researchers did research on ferrate, and we name it activated ferrate 6 oxidation process. This resulted to enhance oxidation of organic contaminants around 30 to 40%. And this happened at much lower concentration of iron 6 and much shorter contact time. We'll be talking about seconds to minutes versus hours. And the good thing was that the final pH was 7.5, which is in line with the regulatory limits, and it's very relevant to wastewater treatment applications. And then this was these findings they have wider implications in purifying water and wastewater from pollutants. At that time, we really thought that other researchers may explore this more. We thought that there is a new field to investigate and to try other activation methods with the goal to produce those iron 4 and iron 5 species, and then to use them to oxidize organics or to oxidize inorganics or for disinfection and many other applications that we could try, which this actually happened with other oxidation processes like photocatalysis, Fendon, and stuff. For example, there is lots of work going on with catalysts for Fendon processes, and there is an enormous amount of studies on the development of photocatalysts to use in heterogeneous photocatalytic processes, and then these findings here, they open the field for investigating activation methods for iron 6 to degrade several other pollutants and in different applications. And of course, this would go even beyond the wastewater treatment, but having an environmentally friendly oxidant, we could use it to produce chemicals in a more environmentally friendly way, and by producing chemicals in green chemistry and in green way, this is also a contribution to water, because we produce less polluted water. So it's really we've been very excited, and we thought that this can be applied in many different aspects. And soon after this finding, I presented this work in the American Institute of Chemical Engineers at a 2016 annual meeting in San Francisco, in the session of liquid phase reaction engineering, and this work was awarded with the best presentation of the session, and that was really satisfying, because going through all this to figure out this to answer this scientific question was really exciting. And then when this is appreciated, it was really something good. And me being a PhD student by that time, you can imagine the feelings, and this resulted to an invitation to publish some work related to acid activated iron 6 to the AICG journal, and this is what we did. And then after this finding, I'd like to talk to you a little bit about what happened next and what was the impact. I'm on purpose, I wanted this webinar to focus on the finding and how we designed the experiments, and how we figure out a new oxidation process. I'm sure in the rest of the webinars there will be a lot of presentations related to how we expand an oxidation process into different pollutants and different ways, and I think those fit very well with this presentation that shows how we could find something new, a new oxidation process, and then go on with expanding this research to different pollutants and different activation methods. And this is what I'm going to talk to you the next 10 minutes, the last 10 minutes of the webinar. So after the first thing we did is that we developed some empirical models using response surface methodology, and I'm sure many of you are using this in your research on oxidation processes. Here on the left side, there are 2 examples. As I showed at the beginning, researchers were very familiar by that time with the molar ratios of iron 6 to pollutant, and we suggested another molar ratio that was the acid or the activator over the iron 6 concentration, and we developed equations that we could predict the removal of a specific pollutant using different activators. Then we started investigating the effect of ions with and without acid, and basically with iron 6 and with activated iron 6, and we've been using different compounds, and some of these findings here they resulted into independent studies. For example, here we can see a little bit of enhancement with the bicarbonate in the presence of bicarbonate, and this later on was used by other researchers in a different study to show that bicarbonate at specific concentrations may also activate iron 6. Of course, we've been very interested in real wastewater, so we did a collaboration with the Agriculture and Agri-Food Canada by that time, and we sampled secondary effluent wastewater from activated sludge-based wastewater treatment plants in London, Ontario, and we treat them with iron 6 and acid-activated iron 6 at the natural concentration of the pharmaceuticals, which was nanograms per liter. And here we can see, for example, in the case of azithromycin, there is a significant enhancement, and this shows that, for example, we need half the concentration of iron 6 to remove azithromycin if we activate iron 6. Here there is an example of sulfametazine that we can get almost complete removal with acid-activated iron 6, but partial removal in the case of no activation. Of course, in some cases, iron 6 alone was also effective, but in this study we could show that we could show two things: that the acid-activated iron 6 could also be applied in the real wastewater matrices, and that, of course, the concentration and the contact time may be important in optimizing depending on the wastewater matrix quality. And in the same study, we investigate few pesticides, and here what was really interesting is that if we see the figure B, and these two pesticides right here, there was almost no oxidation by iron 6 allowed, but when iron 6 was activated, we could see some degradation of these compounds in the real secondary effluent wastewater. So that was the first time that the acid-activated iron 6 was applied in real secondary effluent wastewaters to remove contaminants of emerging concern at their natural concentration of nanograms per liter. Of course, as I said, another important thing was to expand this into different activation methods, and what we showed with the acids was homogeneous reactions, and we also tried some heterogeneous reactions. Here we use silica, which is a solid material, and we could achieve enhancement in the degradation of this model compound. Again, in this case, we use caffeine. We could see that silica alone could not remove caffeine. Iron 6 could partially oxidize caffeine, and then in the presence of silica, we could see a catalytic reaction resulting in almost complete removal of caffeine. And here is one of the figures of this work, where we studied a different concentration of this solid material, and we could see that with 1 or 2 grams per liter, we could get significant enhancement of the decomposition of this model compound. And of course, silica alone, doing control experiments and everything, we could prove that there is a synergistic effect between silica and iron 6. In that time, that was the first activation method of iron 6 using a solid material. And in that case, we went through some identification of oxidation products and reaction pathways. And here we showed that iron 6 attacked the C4-C5 bond of the caffeine molecule, and resulting in these oxidation products. And what was very interesting is that we get the same oxidation products with and without the activation of iron 6 by silica gel, which also supports the initial conclusion that is the iron-based species that are responsible for the observed enhancement. Otherwise, if we had, for example, hydroxyl radical, we would expect some difference in the oxidation products, but this is not what we observed. And in that case of silica gel, which is a solid material, again, we applied that in real secondary effluent wastewater, and here are the results. There are some interesting findings, like in the case of azithromycin, we can see that it could be partially removed by silica alone. There was almost no reaction by iron 6, and then there was an enhancement with the combination of the two. In the case of sulfametazine, silica could not absorb this pharmaceutical. Iron 6 could achieve around 50% removal, and then there was a significant enhancement by iron 6, and similarly in the case of pesticides. And what is interesting here is that we are talking about oxidation and absorption in a single unit, because those silica and iron 6 can be mixed in a single reactor, so we don't need to have a treatment train, or we don't need a series of reactors. Everything can happen in one tank, and we can get several processes: adsorption, oxidation, and depending if we are interested in the activation of microorganisms, then disinfection as well. And other activation methods that we recently found was with Hydrochar. This was some work that I did it was a work that did during my postdoctoral fellowship at Texas A&M University, and then we also found the activation with the peracetic acid and other oxidants, And what was another interesting fact about this finding was that we would be able to interpret some disinfection data that we wouldn't think this way without knowing the activation of ferrate and the formation of iron 4 and iron 5 as the species responsible for enhancing the degradation of pollutants. For example, here is the inactivation of murine norovirus in phosphate buffer and wastewater, and here we can see that we get higher inactivation in wastewater compared to phosphate buffer, which this is it's a surprise, and it would be very difficult to interpret these results. But knowing that iron 6 can be activated and form iron 4 and 5, which are more reactive than iron 6, then this is a way we interpret this result of higher inactivation in wastewater compared to phosphate buffer, for example. So what was expected actually happened, that the activation of ferrate initiated a lot of a great scientific interest by other research groups. Here I'm showing just few studies published from 2017 that it was a discovery of activated ferrate to today. And here you can see some very common words of using the word activating iron 4, iron 5. People found that ammonia activate iron 6. They use carbon nanotube, and they show the involvement of iron 5 and iron 4 species, and then several others. And this resulted in a very recent critical review that I had the opportunity to participate, and this will be available online soon. I guess is accepted by Environmental Science and Technology, where we really review critically the advancement in the activated ferrate 6 oxidation process, and there is a table with around 20 different activation methods of iron 6 the last 4 or 5 years, having acid activated ferrate 6 as the first reported for environmental applications. So, so the main conclusions for of this webinar is that some unexpected results led to a new oxidation process, and this conclusion is I think is especially important for the PhD students, that sometimes unexpected results may lead to something new, and this is when the things are really exciting. And but of course this requires a lot of work and going through the investigation and to prove the findings. And the activated ferrate 6 oxidation process initiated scientific interest, and this resulted to several activation methods by different research groups around the world. And of course, there is lots of work to be done in the future. All these activation methods has to be evaluated in real wastewater matrices, and then to investigate the effect of the water matrix, etc. And of course, to understand the detailed activation mechanisms. There is no method today to measure directly iron 4 and iron 5. So basically, this is something that is gonna we hope is gonna be done the next 10-20 years. And of course, there is work going on regarding the commercialization of ferrate and how to address challenges of ferrate with regards with the stability in different water matrices. So here are some references that I use in my presentation. Here are some contributions, starting from the first one in 2017, going all the way to that critical review in 2022. And I would like to thank my PhD supervisors, Dr. Ejo Ere and Dr. George Nakhla from the University of Western Ontario. As I said, this work was done at the University of Western Ontario with the funding of NSERC, the governmental agency, and the collaborator, Dr. Virender Sharma, Texas A&M University, and also was my postdoctoral advisor. Dr. Minbao Feng for Xiamen University. We've been postdocs together at Texas A&M. The Agriculture and Agri-Food Canada for the work in the real secondary effluent wastewater. I'd like to thank Dr. Luigi Rizzo and my supervisor, Dr. Despofat Takasinos, for giving me the opportunity to give this webinar today, and Dr. Kobe Garavia for being the moderator of this webinar. Before to thank all of you for being here today, I would like to remind you about the 5th Ibero-American Conference on Advanced Oxidation Technologies, which is going to held in November 2022. If you need more information, you can contact Vidor Vilar. Here is the email, and just keep a note that one of the invited speakers is going to be the Professor Despofat Takasinos with a presentation on advanced oxidation process. So I would like to thank all of you for attending this webinar today, and I will be more than happy to respond to any questions from you. Thank you very much.
Thank you very much, Kyriakos. Very nice presentation indeed. So we have one question and two congratulatory comments for you. The question is whether ferrate must be prepared in situ and the lifetime and also the cost per ton of its production.
Yeah, that's actually a very good question. The biggest challenge on the Ferrate 6 technologies is the stability. Ferrate is not stable, so it's stable only for a few hours. So it cannot be used like chlorine, for example, that we can have a barrel and it can stay there for months. Ferrate is not stable, so either we should go with the solid Ferrate 6 with the powder, which this has other implications in producing powder iron 6 instead of liquid iron 6. Regarding the price, this is something that really there is no an answer for that. There is one company producing iron 6 in Texas in a solid form, and there are other people working on producing iron 6 solution. It really depends on how we produce the iron 6 regarding the price. But what I can say is that is competitive to ozone and other oxidation processes, but of course it's more expensive than chlorine in a case that we are talking about application of iron 6 for disinfection of secondary effluent wastewater. But these are the biggest challenges in the iron 6 technology. One is the stability, and the second is that lower stability sometimes results in higher costs.
Okay, thank you very much. So we have a question from Professor Luigi Rizzo. Yes?
Hi, Kyriakos. Thank you very much for your presentation. Really interesting. And thank you for accepting our invitation for the webinar. Yes, I found your presentation really interesting. This is a quite new field for me. But I was wondering, what about the fate of the residual iron after treatment? We need a post-treatment to remove iron in some way, take into account that some regulation have you should meet the compliance with a residual iron concentration in the effluent or eventually for reuse.
Yeah, hi, Dr. Rizzo, and thank you for the question. Also, Despo says hi to you and to everybody. She would love to be with us today, but she has a council meeting and that was not possible, unfortunately, but she says hi to everybody. Thank you, Dr. Rizzo, for the question. Actually, depending on the regulations, iron 3 could be, because it's in the study, in the water treatment pH, iron 3 will be particulate, so it can be removed with different ways. There are people investigating magnetic separation or simple coagulation or simple settling or filtration, depending if the amount of iron 6 that is used is high, and then this is a requirement. But one of the vision of many people that do research on iron 6 is to recover the iron 3 after the application of iron 6 and then use it either as a coagulant in the primary treatment in a wastewater treatment plant, or use it to produce iron 6 again. So this is the vision to recycle iron in a wastewater treatment plant and to avoid the extra step of removing the iron 3 without any use of the iron 3 at the end of the treatment.
Okay, thank you very much.
Thank you.
So any other questions from the audience? All right. If there are no more questions, I would like to thank Kyriakos very much for the very nice presentation. And we have another question in the chat. Actually, I got a notification from Teams that we only have a few minutes left, a couple of minutes left. So the last question is, can activated iron remove phenol from wastewater, and the excess iron sludge can be removed after treatment?
Yeah, so regarding the sludge, because iron is not toxic, when it goes to the sludge, then the sludge does not require any specific treatment. It can be treated as a non-hazardous sludge. If it depends on where the iron 6 is going to be applied. In this case, we are talking about application in the primary treatment. Then if there is iron 3 in the sludge, then this sludge can be treated equally as the sludge that is produced when we use iron 3 as a coagulant in a wastewater treatment plant. And regarding the phenol, yes, there are I didn't work on phenol myself, but there are several studies that show that iron 6 is effective in removing phenolic compounds in water and wastewater matrices.
Thank you very much. So, if there are no more questions, I would like to thank you very much for your presence today in this webinar, and I would like to say that we look forward to seeing you the next webinar in January of the International School of AOPs. Thank you very much, everyone. Thank you, Kyriakos.
Thank you.
Bye-bye. Thank you, Kyriakos.
Have a nice Christmas, everyone.
Thank you. Bye-bye.
Thank you.
Bye-bye.
Bye.
Thank you. Bye.